Good afternoon, students. Today's topic of lecture is upper respiratory tract infection, and one of the commonest organism causing upper respiratory infection in children, diphtheria, in previous days. Now, due to the vaccination, diphtheria cases are very rare. But these are the commonest pathogen associated with upper respiratory tract infection, and that is today's topic. Aim and objective of the lecture. At the end of this lecture, student able to list the organisms causing upper respiratory infection. What are the differential diagnoses? If the patient coming with sore throat, running nose, problem in deglutition, fever, hoarseness of voice, so there is a chances of involvement of upper respiratory infection and may be caused by some microorganisms, either bacteria, viruses, or fungi. Depending on clinical presentation and investigation, we have to diagnose to proper management of the patient. Now, upper respiratory infection in viruses, routine corona and COVID-19 also involvement of upper respiratory tract and also involvement of lower respiratory tract, like pneumonia or pneumonitis. Some organisms only cause upper respiratory infection. Some organisms cause lower respiratory infection, like tuberculosis, and some organisms may start from upper respiratory tract and they spread to lower respiratory tract. But at least we know the list. What are the organisms associated with the upper respiratory tract infection commonest? Then at the end of the lecture, student able to explain pathogenesis and laboratory diagnosis of diphtheria and prophylaxis of diphtheria. Important questions may ask on this topic. LQ may be direct on diphtheria, enumerate the organisms causing upper respiratory infection, describe pathogenesis, complications, and laboratory diagnosis of diphtheria. It's straightforward question. But nowadays, maybe CBME pattern may ask clinical case and then they put the question that I will discuss later. Sharp notes may be asked SAQ, Elix precipitation test, morphology and cultural characteristics of Corynebacterium diphtheria, DPT vaccine. Such a case may be asked in exam and then how to they ask the questions. This is the partially immunized 8-year boy developed sore throat. It's a case of upper respiratory infection. On examination, patient is toxic, means upper respiratory infection with toxemia, and pseudomembrane is seen over the tonsils and pharynx, facial area involvement. This case given also asks the question, what is your diagnosis? Then etiopathogen laboratory diagnosis of this case, prophylaxis of this patient, and treatment or management of this patient. Sometimes we give the diagram and investigations of gram stain. The throat swab may show gram positive bacilli. On this investigation and clinical case, you have to find out the positive agent of the patient among differential diagnosis. Now, the first case is partially immunized. Then clinical cases I put and I will explain in end of the class. Partially immunized and patient is toxic. What is the clinical diagnosis? In second case, the child is immunized and in non-toxic. So what are the possibility? And again it is a gram positive bacilli infection. What is the possibility in immunized person having the sore throat, pharyngitis or upper respiratory infection, but patient is non-toxic? The question may ask why we can't prevent carrier stage by vaccination. Maybe we can't prevent the infection and we prevent the disease by DPT vaccine. So what is the mechanism? Okay. So what is upper respiratory tract? Upper respiratory tract already we know anatomy and physiology. These are the parts of nasopharynx, nose, mouth, sinuses, trachea, bronchi, lung, and larynx. They depend on the site of infection. Why the different organisms have different site? We can depend on receptors on a particular site and the involvement. So maybe it is patient has sinusitis, common cold rhinitis, pharyngitis, nasopharyngeal infection, tracheitis, epiglottitis, tracheobronchitis, only bronchitis, and pneumonia and pneumonitis, lobar pneumonia and interstitial pneumonia, patchy pneumonia, atypical pneumonia. There are different sites and different organisms involvement and terminology. So that is not I'm going in detail in each and every because our part is today that is diphtheria. So if you know what are the upper respiratory infection and what are the different types of infection, common cold it may be caused by viruses like corona, COVID-19, adeno, influenza, and maximum rhinoviruses. Running nose. So sinusitis associated with the common cold. Pharyngitis and laryngotracheitis. Laryngoepiglottitis, also known as croup, sometimes with acute condition, is caused by Streptococcus pyogenes, Corynebacterium diphtheria, Haemophilus influenzae type B, most common, many viruses, also parainfluenza viruses. Epiglottitis, Haemophilus influenzae type B, or other also. Bronchitis, Bordetella pertussis, and many other viruses like respiratory sensitive syndrome or respiratory sensitive virus, RSV. So these are the commonest organisms and commonest site as overlooked upper respiratory involvement by different microorganisms. The today topic is Corynebacterium diphtheria. Diphtheria. Diphtheria, it is disease mostly occur in the throat. Commonest site is fossil diphtheria and commonly occur in children. But nowadays we are rarely cases of diphtheria due to National Immunization Schedule, involvement of DPT vaccine, awareness, and every child getting DPT vaccine. The severe form of diphtheria, we are not getting cases. And routine treatment, we can treat the patient by giving simple penicillin or erythromycin antibiotic. So DPT vaccine is mass vaccination, involvement in national immunization schedule, the diphtheria cases are very rare. Okay, so these are the points those we are going to discuss in the class. First, causative organism Corynebacterium diphtheria, how it looks, what are these properties. Second, etiopathogenesis of diphtheria. Third, laboratory diagnosis of diphtheria. And last one is prophylaxis of diphtheria. The Corynebacterium diphtheria, causative agent of diphtheria, mostly commonest site is throat or fossil or tonsils, but it may be caused nasal diphtheria, conjunctival diphtheria, otitic diphtheria, genital diphtheria, cutaneous diphtheria, laryngeal diphtheria. But commonest site is fossil diphtheria, throat, pharynx. In morphology, most commonly staining method is gram stain that is used for most of the bacteria. It looks like gram positive bacilli, non-sporing, non-motile, non-capsulated. In gram stain, it looks in arrangement characteristic that is known as Chinese letter pattern arrangement or cuneiform arrangement like V, T, L shape. They not detached from each other and they present in unite each other like V form, X form, P form. They attach each other. So that is gram positive bacilli. And in Albert stain, a special staining method we use to demonstrate Corynebacterium diphtheria in throat swab. Because characteristic feature is that this pointed like structure, these are not spores, these are the granules, metachromatic granules, and that is the characteristic morphological appearance of Corynebacterium diphtheria. It is also known as Unbab's granule, polytin granules, and they present polar bodies. So these metachromatic granules, special staining method is toluidine blue. And why the name is metachromacia? Metachromatic due to metachromacia. We are staining by the toluidine blue. That is one of the stain in Albert stain, Ponder stain, and Neisser stain. Albert is the name of the scientist who discovered this staining method, including toluidine blue O. The color appearance of the stain is blue, but after staining, spores looks like reddish purple color, and that term is known as metachromacia. So it is known as metachromatic granules. It is made up of polymetaphosphate. So this is the characteristic features of identification of causative agents. So Albert stain is used for the Corynebacterium diphtheria to stain metachromatic granules. Another important point is Chinese letter pattern or cuneiform arrangement. They arrange in V, T, L, and M shape. They attach each other at the end. So these two property is used to diagnose the case when we suspect pharyngitis. So this is the morphology of bacteria. Now cultural characteristics. The media used for isolation, one is the enriched medium that is low flux serum slope, and second medium is used that is the blood agar. potassium telluride blood agar. Local serum flow is enriched medium. It is containing serum, so it is enriched medium. And advantage of this medium, the colony appears in 6 to 8 hours, rapid growth. So this medium is used for early isolation of the bacteria, and colony appear in 6 to 8 hours. Another property of local serum slope, granules appear very nicely when we inoculate the specimen in this medium. The throat swab inoculate into local serum slope and incubate for 6 to 8 hours in 37 degrees Celsius, and we observe the growth. Another medium, that is a potassium telluride blood agar. It is a selective media. It containing telluride. It inhibits the growth of other unwanted bacteria and enhances the growth of Corynebacterium diphtheria. So it is a selective media. And colony appear a black color colony due to reduction of telluride in tellurium. But it takes 2 days. 48 hours need incubation, and negative report only after 2 days incubation. So these are the two media mostly commonly used isolation of Corynebacterium diphtheria routinely, that is local serum slope, early diagnosis, and potassium telluride blood agar is selective media, selective growth, when it takes time, that is disadvantage, 48 hours. Hollies and MacLeod's media also used for isolation of Corynebacterium diphtheria. Third important point, that is a virulence factor, that is diphtheria toxin. Diphtheria toxin, that is one of only the complications of diphtheria caused by diphtheria toxin, and the term is known as toxemia, not a bacteremia. Bacteria not enter into the blood. Primary infection in the pharynx or throat. These bacteria secrete the toxin, and the toxin enter into the blood. The term is known as toxemia. And all it is exotoxin, gram-positive bacteria, so it is exotoxin. And all the complications, systemic complications associated with the toxemia. So what is the toxin? Known as diphtheria toxin. Property, it is exotoxin. Gram-positive bacteria only produce exotoxin. Protein in nature. And having two units, A and B. B for binding, A for enzymatic action. B point. Binding is very important. Target site. Without binding again depend on the receptor. Receptor present particular site, so toxin act on particular site, and that is specific action, not a generalized action. So diphtheria toxin have specific receptor on particular organ, so that organ only get involved. So property, it is exotoxin, specific action, binding according to receptor, and A for enzymatic action, mechanism of toxin. Factors associate the toxin production. Again, two important factors of the toxin production, diphtheria toxin by Corynebacterium diphtheria. One is critical concentration of iron. 0.01 unit per ml of iron, or whatever be the unit concentration of the iron. It is the stimulate that gene produce the toxin. Iron level either more or less, there is no production of toxin. The critical level of iron is important factor to produce the toxin. Second, Corynebacterium diphtheria actually is a non-toxigenic bacteria. One in infected by phage, specific phage, either beta phage or tox phage, then bacteria become toxigenic. That is known as lysogenic conversion. The phage infected bacteria only associate the produce the toxin. That is known as lysogenic conversion. The information of toxin production, that gene present in the virus means phage. And then virus when infect coroniphage or coroniphage, then when infect to the Corynebacterium diphtheria, then this virus genome integrated with the bacterial genome, and that term is known as lysogenic conversion. Then this bacteria is able to secrete the toxin. Otherwise, Corynebacterium diphtheria mostly non-toxigenic. Only infected bacteria is toxigenic. Phage infected bacteria. The two factors are important. Bacteria should be infected by phage, known as lysogenic bacteria, and second is the critical concentration of iron. Now action, detection and toxin. The action with target sites. Again, I'm telling target sites, it depend on the toxin having a specific receptor particular site. Diphtheria toxin having the receptor on the myocardium, so toxin is attached to the myocardium and cause cell death, myocarditis. And according to myocarditis, there is a cardiac output is decreased, patient is shock, and patient is circulatory failure. So oxygen supply to the vital organ is decreased, and patient there is due to the death due to asphyxial damage of the internal vital organs like kidney, acute tubular necrosis, and brain due to ischemic damage of the brain coma. Detection, again it's part of laboratory diagnosis, how we detect the toxin, that I will tell in detail. These are the properties, two fragments A and B, B for binding, this is B for binding, and A for enzymatic action. They bind to the particular receptor, A for enzymatic action. And what is enzymatic action? It blocks the protein synthesis. It blocks the protein synthesis. A block the protein synthesis, prevent the elongation factor 2, which affect the protein synthesis, ribosomes. In ribosomes is block elongation factor. And due to block the elongation factor 2, there is no production of protein. So decrease the protein synthesis or suppress the protein synthesis and then the cell death. Inhibition of protein synthesis by inhibiting elongation factor 2. Okay. Again, toxide, it is used for vaccine preparation, and that's why we use DPT. DPT means toxide. Particularly diphtheria toxide, not toxin, and tetanus toxide. So we use toxide for the vaccine. Toxin having a pathogenic or virulence property, toxide, we remove the virulence property and maintain the antigenic property to stimulate the immune system. The toxin convert into toxide by treating the toxin with formalin, prolonged storage of toxin, incubate for prolonged time, and alteration of the pH. Toxin convert to toxide. And toxide is immunogenic, that able to stimulate the immune system with production of specific antibody against the toxin products. But virulence property we destroy, and that is known as toxide. Toxin with virulence property, toxide without virulence property. So what is the importance of toxide? We can use for vaccine preparation. How you prepare toxide from toxin? Treated with the formalin, prolonged storage, and alter the pH. Two fragments. Again, this B I am telling. When we use the toxide, when we stimulate the immune system, the antibody form against B is very protective compared to A. A actually it is a proper action. What are the mechanism? Elongation factor inhibition by A. Protein synthesis blocked by factor fragment A. Cell death, all the mechanism due to factor A or fragment A. When B is the binding. But without binding, A can't enter into the cell. Without binding, A can't enter into the cell. So when they prepare the antibody against fragment B, it blocks the actually attachment of the toxin to the target site. Once there is no attachment, so fragment A not enter into the cell. This is the first part regarding to the positive agent. What is the morphology, Alberstein and Gramstein appearance with diagram must know. Cultural characteristic, what are the media we are using to isolate Corynebacterium diphtheria, low serum slope, and potassium tellurite media, advantages and disadvantages. Third important part is the toxin, because it is the only all the complications associated with the toxemia, and our aim of the vaccination to prevent the toxemia. That's why we are giving the toxide. We are not preventing the multiplication of bacteria and the local infection at throat. We are only preventing toxemia. So now etiopathogenesis. Pathogenesis, these are the points we how to discuss in pathogenesis of diphtheria. One is source of infection, mode of transmission, route of entry, virulence factor, agent host interaction, incubation period, clinical presentation, and complications. The source, these are the mostly the carriers. Right? 3 to 4% people are carrier. I mean the outbreak in the society, and mostly nasal carrier and pharyngeal carrier. Nasal carrier bacteria present prolonged in the carrier space compared to pharyngeal carrier. It is transient, not for prolonged time, 1 year, 2 year, but it is a transient carrier, nasal carrier and pharyngeal carrier. Patient also source of infection. When they cough or sneeze, cough and sneeze, The bacteria spread in the air and through inhalation they enter into the body by mode of transmission. They are cutaneous diphtheria also. So skin also, that is the source of infection. Other parts of the source of conjunctivitis, if there is a conjunctival diphtheria, nasal diphtheria, or otitic diphtheria, laryngeal diphtheria, genital diphtheria, and cutaneous diphtheria. But most source is the cutaneous diphtheria exposed to the other. And another most common site is facial diphtheria, cough and sneezing, that is spread the infection in the air. And through inhalation, they enter into the body. Mode of transmission through air inhalation. Incubation period, virulence factor, in agent host interaction, and the incubation period. The virulence factor I told are diphtheria toxin, and they multiply in the throat, other parts, and they secrete a toxin. Toxin enter into the blood, toxemia, and then target site is most important is myocardium and cause myocarditis. Incubation period is 3 to 4 days, not more than week. It is less than 1 week, and clinical presentation depend on the site of infection. So now the commonest site is the sore throat. So cough, hoarseness of voice, pain in deglutition, fever. That is the commonest presentation of the patient. If the patient also the toxemia, then patient is toxic. And it is in the myocarditis, then patient is circulatory failure. It is low. And characteristic again of this infection or diphtheria is a formation of pseudomembrane. How the pseudomembrane is formed? When bacteria multiply this site, they secrete a toxin. And due to secrete toxin, surrounding cells get necrosis. Due to surrounding cells get necrosis, there is an acute inflammation. Due to acute inflammation, there is infiltration of the neutrophil, other cells, and exudative material. And all this combined, necrotic tissue, matlab cell death by protein synthesis, surrounding tissue cellulitis, cell death, that necrotic tissue, then inflammatory cells, exudative material, and multiplying bacteria, they form one layer. They form leathery layer. So then diphtheros means layer. They form one leather-like layer, and that it is pseudomembrane formation. This is a cutaneous diphtheria. It's mostly caused by non-toxigenic Corynebacterium diphtheria, only by due to the multiplication and destruction by the inflammatory response. Okay. Depend on the site of infection of presentation, they classify into 3 groups. One is a malignant infection, septic infection, and hemorrhagic infection in the sore throat. If there is also the bleeding, suppose we remove it is the site of bleeding, that is a hemorrhagic diphtheria. Necrotic tissue called cellulitis, that is septic diphtheria. And patient also the toxic with severe adenitis, like this. This is the adenitic gland, and this is a bull neck appearance. So suppose this presentation that is known as the malignant or hypertoxic complications of diphtheria. The involvement of the adenoid gland and the neck is become bull neck. This is the case is a laryngeal diphtheria. So what are the complications? One is the asphyxia. But the asphyxia, because this layer, if you're not treated properly in early stages, so this layer they block the airway. When you spread to the trachea or again below the pharynx, then they block due to layer by layer and layer, the airway become obstruction. And involvement of suppose in larynx, then again there is a chances of laryngitis and laryngeal diphtheria, so airway obstruction. Asphyxia is a mechanical complication. It is not due to the toxin. It is due to the multiplying bacteria, and there is a layer formation, and due to layer there is asphyxia. So it is a mechanical complication. Other complications, these are due to the toxemia. When the bacteria secrete a toxin, the toxin enters into blood called toxemia, and again the commonest site I know it is a myocardium. So of the acute circulatory failure. It may be act on ciliary and nerve. palatine and ciliary nerves involvement, and there is a paralysis of that particular muscle supply by this nerve, that known as post-diphtheritic paralysis. When it is recovered, it is reversible. The involvement of most commonest nerves are palatine nerve and ciliary nerve. That is one of the another complication, neuritic complication of diphtheria. Circulate complication, neuritic complication. Septic, due to already I told you there is a cellulitis surrounding the sore throat or membrane, and that necrotic tissue, also the pseudomembrane, that was septic diphtheria. Bull neck involvement of the adenoids and hypertoxic condition, malignant condition. It is a severe condition. In medical emergency, diphtheria is one of the medical emergency. Laboratory diagnosis only for the confirmation, not for the treatment of the patient. It is a medical emergency. And laryngeal diphtheria, it is also one of the medical emergency and need tracheostomy, urgent tracheostomy because asphyxia. The complications, this is a separate slide again. Systemic complication due to toxin. Toxin diffuses into the blood and causes toxemia. Again, I'm telling no bacteremia caused by Corynebacterium diphtheria. Toxemia, good example is Corynebacterium diphtheria. Myocarditis, because target site is the myocardium. Cell necrosis by protein synthesis. Due to cell necrosis, there is a myocarditis inflammation, and due to myocardium involvement, there is no pumping of the heart. So no pumping, cardiac output is decreased, patient in shock, and that will have acute circulatory failure. Internal organ hypoxia and patient death. So it is medical emergency. Now clinical case, before we start the laboratory diagnosis, already I told this clinical case. Now this is the clinical case. Suppose to confirm the etiology, we go for laboratory diagnosis. Laboratory diagnosis, not for other investigations I am telling here, only microbiological investigation. But again telling diphtheria is only the clinical diagnosis to management of the patient, and laboratory diagnosis only to know the etiology. It is epidemiological purpose. How many cases in the society caused by diphtheria? What are the etiopathogenesis or etiology of upper respiratory infection? Upper respiratory infection, how many cases caused by diphtheria? So for epidemiological purpose, need the confirmation of the diphtheria, we need laboratory diagnosis, microbiological investigation. So these are the points we go discuss in laboratory diagnosis. One is the collection of specimen, transport the specimen, microscopy, isolation, identification of species and biotypes, detection of virulent strain, means detection of toxin. The toxigenic strain hai या non-toxigenic strain hai. Already that told detection of toxin, diphtheria toxin. The detection of toxin we will discuss in laboratory diagnosis. Collection of specimen, mostly throat swab. Commonest site, we depend on the site, genital swab, conjunctival swab, otitic swab, and nasal swab. So these are the swabs we have to collect, but common site is fossil diphtheria. The pharyngeal swab, we have to collect 2 swabs, one for microscopy, one for culture. Without touching to the mucosa or saliva, with the membrane, we should rub the membrane thoroughly, and from the membrane, we have to collect the swab, not from other site. And when go for microscopy, one the gram stain and Albert stain. Gram stain, we observe the gram-positive bacilli arranged in Chinese letter pattern. It is the confirm it is the case of Corynebacterium diphtheria, because not any gram-positive bacteria are in Chinese letter pattern in throat infection other than Corynebacterium diphtheria. So gram-positive bacteria arranged in Chinese letter pattern, it is a mostly non-sporing, non-motile, and non-capsulated. This is the characteristic features of Corynebacterium diphtheria. But to metachromatic granules to see in properly, the other stain is Albert stain, Ponder stain, and Lizard stain. The Albert stain containing toluidine blue O, and that staining method where the malachite green and toluidine blue O, 2 stains are there. Malachite green stain the bacilli and toluidine blue stain the granules. So red purple color granules against the greenish bacteria we appear in the Albert stain and maybe greenish background because malachite green. So this is the special staining method for Corynebacterium diphtheria to detect the metachromatic granules. It is a rapid diagnostic method. Within half an hour, we report the physician. These are the gram-positive bacilli arranged in Chinese letter pattern, suggestive of Corynebacterium diphtheria are present in gram stain. Albust and metachromatic granules are present, and green colored bacilli suggestive of Condobacterium diphtheria arranged in Chinese letter pattern are present. This is the suggestive of, not confirmed, still by culture. Then locus serum slope within 6 to 8 hours we get the growth, and growth is confirmed by biochemical reaction and microscopy. Potassium tellurate media also confirm the etiology, but it takes 2 days to give, and black color colony due to the reduction of tellurite into tellurium. I'm not going in detail in cultural characteristic and biochemical reaction. The three biotypes depend on the presence of granules and cultural characteristic and biochemical reaction. The depend of the biochemical reaction, arrangement of the granules, cultural characteristics. There are three types, but these names depend on the severity. These three strains. So which are the strain associated with the severe complications, hemorrhagic and nephrotic complications. So that is known as gravis. Those strain mild complications, not severe complications, so known as mites. And those are in between, not gravis and mites, that is intermediate. All the three produce the toxin. But toxide, to produce the toxin, when we use for toxide, that is the Wark William 8 strain to prepare the toxide. And it is produced by mites strain and not by gravis strain. Now detection of virulence strain. Now we collect the sample, throat swab, we isolate the growth in low serum slope and potassium tellurate media. Within 6 to 8 hours, we observe the growth and low serum slope. Now it is a toxinic and non-toxinic strain. That for we perform the virulence test for virulence, detection of virulence strain by Elixir precipitation test. That is one of the vitro tests we use to detect the virulence strain. Means the detection of toxin. Isolated strain is toxinic or non-toxinic. It is a phage infected or non-phage infected. Elixir precipitation, it is important short note. This is the diagram you must have to draw with label. Elixir precipitation test, that is the media we use, it is 20% horse serum agar. And one square shaped strip you are observing this is the filter paper soaked with diphtheria antitoxin. Then in the center, we inoculate the control strain, positive control strain. And in the right side, there is a negative control strain. This is the positive control strain left side and negative control strain right side. Negative control strain matlab means non-toxigenic strain. Positive control, toxigenic strain. And in the between, that is the test strain. I don't know it is toxigenic or non-toxigenic. It is a isolated strain, clinical isolate from the patient. This is the positive strain. This is a negative strain, toxigenic. And this is an isolated strain from the patient. Now, if it is a toxigenic, it forms the line of precipitation. Why line of precipitation? These bacteria present in this isolated or positive strain produce the toxin, and antitoxin from the filter paper is diffused into the medium. Then the antitoxin toxin get combined. If it is homologous, they form line of precipitation. And both toxins are same if they form the toxins line of precipitation toward the one direction. They unite each other. Once they one direction unite, that is the same strain or same toxin produced by the isolated strain. The isolated strain is toxigenic. If it's non-toxigenic, there is no line of precipitation. The non-toxigenic. This is simple test, Elixir precipitation test, and 20% horse serum agar. One, it is a filter paper soaked in antitoxin placed in the center of the medium. Positive negative control perpendicularly inoculated on the media, and isolated strain from the patient inoculated in the center of the medium. Then we incubate in the incubator for 1 day or room temperature. Then the bacteria they multiply and they produce the toxin, and antitoxin diffuse from the strip, impregnated strip, and if they are both are homologous, then they form the line of precipitation. This is precipitation in gel-like reaction, so Elixir precipitation test. Other also test in vivo test, guinea pig we use, but now animal experiment outdated. So another is tissue culture test also there. This toxin destroys the tissue. The monolar of the tissue we prepare hero cell or viro cells. Isolate organism from the patient in inoculate the tissue culture bottle, and the cytopathic effect observed under the microscope. If the monolar get disturbed or destroy the cell, that is the toxigenic strain. That is known as tissue culture test. This is a elixir precipitation test. Treatment of the patient and treatment of the carrier. Carrier are contacts. When the patient, घर में एक patient है, उसके बाद में सभी में अगर उसके दोस्त या उसके family members contact में आते हैं, तो then we have to treat also them to prevent the carrier. They don't have contacts. And if you detect the carriers, also we give the treatment to prevent the spread of the infection in the society. The patient drug of choice is the penicillin, and again we give the ADS, anti-diphtheric serum. In acute cases, with medical emergency, toxin we have to neutralize. Penicillin only kill the bacteria, not the toxin. So toxin we have to neutralize by giving the anti-diphtheric serum. We prepare in the horse. Toxin inoculate to the horse, and we collect the antibody against the toxin from the horse serum, and then passive immunization. And then with the antitoxin 20,000 to 1 lakh unit we can inject if the patient depend on severity to neutralize the toxin and further damage of the target site, means myocardium. That is like passive immunization. It is a prophylactic as well as therapeutic. Therapeutic dose is more, up to 1 lakh. Prophylactic, if it comes in contact with that patient, then you get the prophylactic dose, that is 500 to 1000 international unit, without any clinical presentation. That is passive immunization. And in therapeutic treatment, we do the ADS serum. Drug of choice is penicillin and ADS. That is treatment of the patient. Carrier, drug of choice is erythromycin, then penicillin G. And those person having allergy, mostly penicillin, so many people having allergy. Erythromycin, drug of choice for carrier or contacts. And drug of choice for patient is penicillin G. And those are having the allergy, then erythromycin. Okay. DPT vaccine, that is maybe asked in the exam, prophylaxis. There are 5 doses, 6 weeks, 10 weeks, and 14 weeks. The primary doses, and then booster doses we give second year and 5 year. And DPT, D for capital D for children, dose is more, 20 to 25 Loeffler's flocculation unit, and adult dose is 2 to 5, denoted by small d. The small d for smaller dose, capital D for larger dose. Larger dose for children, primary immunization. And in adult, maybe they are exposed to an environment, so we should not give the full dose. So we give the small dose, 2 to 5 unit, compared to 20 to 25 unit in children. So DPT, diphtheria, pertussis, and tetanus. Diphtheria toxide we use here combined, so there is a enhanced effect of stimulation. So combined vaccine. This is immunization schedule. Primary doses 6 weeks, 8, 10 weeks, and 14 weeks, and first booster second year and fifth year. Fourth dose and fifth dose we can say. This is the name without in diphtheria most before era of antibiotic start 42. Before that he discovered ADS for the treatment of the patient in diphtheria, and that was drastic. He saved so many lives from toxemia by giving simple ADS, and that Nobel Prize awarded, that is wine bearing. So ADS also single day dose, that is the one meaning, because when we use the horse serum, when we give the second dose after some days, there is chances of type 1 hypersensitivity, because horse serum albumin are combined with antibody that is foreign to the patient, and patient may become sensitization to the horse serum. So second dose it causes shocking dose and anaphylactic shock. So single dose, single day therapy only used for ADS. Not repeated doses should not give in the patient. It may be immune elimination or anaphylactic shock. So for his discovery, that time he saved so many lives. That time no antibiotic, no prophylaxis, no vaccine. It's a commonest infection among the children, and due to ADS treatment, he saved so many lives and awarded the Nobel Prize. Now to get the exercise last 2 or 3 slides, because so many people sometimes miss the video, miss the PPT. But to get the attendance, at least you read the chapter today. And if I will get time, tomorrow I will again arrange one Zoom meeting for 10 minutes to give the answer of these questions. If the answer you are getting wrong or right, that it doesn't matter. Just you solve the problems and send to me on WhatsApp. I'm just filling here the questions, and tomorrow I will give the answer. One is partially immunized 8 years boy develops sore throat. On examination, patient is toxic and pseudomembrane is seen over the tonsil and pharynx. Throat swab, gram stain shows gram positive bacilli. What is your clinical diagnosis? The investigations behind पेशेंट का हिस्ट्री भी है, राइट 2 or 3 lines कि why it is a case of diphtheria and why the patient is toxic. Now, another case, it is successfully immunized 8-year boy, developed the sore throat on examination, patient is non-toxic. Immunize है, non-toxic है, same history. And question is, second is diphtheria, no problem, fossil diphtheria. But question 2, you have to explain why we can't prevent carrier stage by vaccination. Again, for this, I am giving the clue here. Now we are getting the vaccine of COVID-19, 2 doses. And after 2 doses, so many people are getting infection. The getting infection vaccine aim here. What is the aim of vaccine here? And what is the aim of vaccine COVID-19? The aim of vaccine at present COVID-19 to prevent the lower secret infection, pneumonitis. Because we are giving injectable vaccine. So injectable vaccine, they synthesize IgG antibody and protective only tissue, not the surfaces. To prevent the surface infection, we need IgA type of antibody, and IgA type of antibody only by live vaccine, intranasal vaccine. It will come in future. The till we can't prevent the sore throat and nasal COVID-19. COVID-19 or corona infection, we vaccinated people also will get, but pneumonitis not develop in vaccinated people. So there is no hospitalization, and still we not hear any death in vaccinated people. The upper respiratory infection we will get after vaccination also, and we can prevent others also. So mask is very important to prevent the transmission and prevent the upper respiratory infection. Vaccination aim at present only prevent the pneumonitis and prevent the hospitalization and mild infection. Still no death in vaccinated people. So after 2 doses, we are getting the corona infection. It is common because we are not protecting the nose and throat. We are protecting lung only at present. But why wear the mask? We are supposed we get the upper respiratory infection, we transmit to others. And virus multiply continuously in the society till it get eradicated. Our herd immunity will increase. And how many times it multiply, it may be chances get mutation. And if with more mutation, then vaccine also may be ineffective. So prevent the transmission is most important role to wear the mask to prevent the transmission, to prevent the chances of multiple cycles of the multiplication of virus. So we prevent the mutant strain in arise in future. And vaccine to prevent the hospitalization. Okay, the same principle also applied here also. Why we can't prevent the fossil diphtheria carrier stage by vaccination being repeated. MCQ about diphtheria true is read this 6 MCQ. PPT I will share and send to me today or by tomorrow. But tomorrow, if possible, I will take 10 or 15 minute Zoom meeting to answer of these questions. Thank you.